Hello everyone. Uh, we will today discuss about a common condition called as xanthoma, and the basic pathology in xanthoma is because of defect in lipid pathways. We'll see how the lipid is being absorbed in our body and what's the pathway and how it leads to xanthoma and xanthelisma and other conditions. I'll be explaining all the pathways with the help of a diagram. First, whenever we have a fatty meal, the whatever the food we consume goes to the stomach and then through the intestine. Uh, when we look at the image, the food which we consume has lipids in a complex form that will be in the form of triglycerides. These triglycerides will enter the stomach. Through the stomach, it reaches the small intestine. In the small intestine, there is secretions of pancreatic lipases and also bile from the liver. This is the gallbladder. Gallbladder will be having concentrated amount of bile, and this is the pancreas. Through the common duct, it gets released into the second part of the duodenum. Here is where the fat absorption starts. First, triglycerides will be broken down into monoacyl glycerol and free fatty acids. Triglyceride contains 3 carbon atoms bound to 3 fatty acids compound. This forms a triglyceride. When pancreatic lipase acts on the triglyceride, it gets broken down into monoacyl glycerol with only one free fatty acid attachment and free fatty acids. So when these monoacyl glycerol and free fatty acids are present in the second part of the duodenum, there is something called as duodenal brush border cells. These duodenal brush border cells helps in absorbing this monoacyl glycerol and free fatty acids. Once they get these dots represent monoacyl glycerol and free fatty acids. With the help of the file, they form a complex form known as micelle. As it is not able to easily absorb the free fatty acids and monoacyl glycerol, it is converted into micelle formation with the help of a bile. This micelle is easily being absorbed by the second part of the duodenum. Once the micelle enters the second part of the duodenum, it goes through the rough endoplasmic reticulum, then smooth endoplasmic reticulum. Before going through the rough and smooth endoplasmic reticulum, it again rejoins to form the triacyl glycerol. After forming triacyl glycerol, it goes to rough endoplasmic reticulum and smooth endoplasmic reticulum. This is rough endoplasmic reticulum, and there will be smooth endoplasmic reticulum. While it passes through, there is a protein called as ApoB48 which is attached to the triacyl glycerol to form a compound known as chylomicron. So chylomicron will be released from the second part of the duodenum intestinal cells into the blood vessels and into the lymphatic vessels through a process known as reverse spinocytosis, that is reverse of endocytosis. Once it reaches the lymphatic system, all the fatty meal will go through the lymphatic system into the thoracic duct, which is the main lymphatic channel, and this thoracic duct will drain into the aorta at the junction of left subclavian vein and left brachiocephalic vein. Once it reaches the circulation, this is the chylomicron. It is called as a chylomicron remnant. This chylomicron remnant will be having ApoB48 attached, and inside there will be triacyl glycerol and cholesterol. This is known as nascent chylomicron. Sorry, ApoB48. This is called as nascent chylomicron. This is nascent chylomicron, and there is a particle called as HDL. This is HDL. This HDL contains ApoC2 and ApoE. This ApoC2 and ApoE are transferred to the chylomicron and then convert the nascent chylomicron into proper chylomicron. So now it contains B48, C2 and ApoE. Why this C2 is important is because in the endothelium of the blood vessels there is an enzyme called as lipoprotein lipase, which is necessary for breakdown of triacylglycerol into free fatty acids and glycerol. This lipoprotein lipase will not be activated usually. It needs presence of C2 to get activated. So when the HDL donates the C2 to the chylomicron, it will be able to be processed by the lipoprotein lipase. This ApoC2 will attach to the lipoprotein lipase like a magnet, and then chylomicron will be broken down into glycerol and free fatty acid. This glycerol and free fatty acids will be taken up by the muscles. This is the muscle, or it can be taken up by the adipose tissue. Two pathways it can enter, or it can enter back into the liver where it will be used for synthesizing the bile. This is about how the chylomicron and how its pathway. Once chylomicron is broken down, it will be converted into chylomicron remnant, and it has this chylomicron remnant will donate the ApoC2 back to the HDL, and it so C2 is here, it is going back to the HDL. So only it contains ApoE and ApoB48 as usual. This ApoE acts like a magnet to the liver, where it will be taken up by the liver cells and broken down to form triacylglycerol and free fatty acids and cholesterol, which will in turn be incorporated into the bile formation. This is how the chylomicron cycle takes place. Now once the liver, here we'll take the liver, this is the liver. Inside the liver there is synthesis of LDL, VLDL actually. In the inside liver there is synthesis of VLDL. So VLDL is synthesized by a combination of triglycerides and cholesterol. Will pass through rough endoplasmic reticulum and smooth endoplasmic reticulum of the hepatocytes of the liver and get ApoB100 to form VLDL. So initially VLDL contains only ApoB100, and that is secreted into the lacteals of the liver, from which it in turn drains into the blood circulation. Once VLDL reaches the blood circulation, initially it will be having ApoB100 only, and here comes our good cholesterol, that is HDL, again containing ApoC2, ApoE. This donates the ApoC2 and ApoE to the VLDL so that it can form proper VLDL with B100, C2 and ApoE. So that now the VLDL will in turn can be attracted to the LDL, VLDL can be attracted to the lipoprotein lipase in the form of a magnet with the help of ApoC2. So VLDL comes to the lipoprotein lipase, gets broken down to form triacylglycerol and free fatty acids to form IDL. This IDL in turn can get converted into LDL. Okay, in this process it will give back the ApoC2 back to the HDL. So LDL will be containing only ApoB100 and ApoE. This ApoE is necessary for the LDL to be taken up back by the liver. Now in all of this process we didn't tell how the HDL was produced. HDL is mainly produced by the liver cells and also by the intestinal cells. This HDL contains an important protein other than ApoC2 and ApoE called as ApoA. This ApoA is responsible for the helpful action of the HDL. So if we look here, this is a HDL containing ApoC2, ApoE and ApoA. So normally there will be chylomicrons, there will be VLDL, there will be LDL. All this contains cholesterol. What this HDL does is it takes the cholesterol from all the three structures in the form of cholesterol. This is the cholesterol, cholesterol, cholesterol. From all the three, cholesterol will come to the HDL. This cholesterol needs to be converted to cholesterol esterase to be stored inside the HDL. For that we have an enzyme called as LCAT, lecithin cholesterol acyl transferase. This LCAT converts cholesterol to cholesterol esterase, and this cholesterol esterase gets accumulated within the HDL, and then HDL can in turn give it back to the IDL so that it can be transported back to the liver, where it can be synthesized into bile and excreted into the feces. So this in turn cholesterol will be given to the IDL. IDL will carry it back to the liver. Liver will synthesize bile, and this bile will be secreted in the feces. So 99% will be absorbed, only 1% of the cholesterol will be secreted in the form of bile in the feces. That is how we lose the cholesterol from our body. So this process of HDL taking the cholesterol from chylomicron, VLDL and LDL and giving it back to the liver is called a reverse cholesterol transport, and the main enzyme responsible for this is LCAT. So in all this pathway where the xanthoma, xanthelisma occur, so what happens with this LDL and all, these are LDL is the most concentrated form of the cholesterol. This LDL and then free fatty acids and triglycerides, they can get accumulated in the tissues such as skin. So this is the surface, this is the skin with the hair. Okay, this is the skin. This can deposit here. When this deposits, there is a army fellow called as macrophage which engulfs this fat and forms into foamy macrophage. This foamy macrophage is what accumulates in the skin, leading to xanthoma formation or xanthelisma formation. These free fatty acids can also accumulate inside the blood vessel, leading to atherosclerosis. So if this is the blood vessel, a plaque can form on the blood vessel, forming atherosclerotic plaque, and platelets can plug over it. Thrombus can form and completely occlude the blood vessel, leading to ischemic attacks. If it occurs in the heart, it's called myocardial infarction, and if it occurs inside brain, it's called as stroke. So this is how the xanthoma formation and lipid pathway takes place, and if there is any enzyme defect, that will lead to various genetic disorders. For example, if there is a, which we'll be discussing, all the genetic disorders we'll be discussing in the next part. Thank you.